My name is Dr. Sigalit Zchut. I work for Enzymotech, and I'm the R&D director. And today I'm going to talk to you about phospholipids, small molecule, big promises, phospholipids, the game changers in the omega-3 world. So who is Enzymotech? Enzymotech is an Israeli-based company. We're located at the northern part of Israel. We are a company that puts all of the emphasis and centralized on lipids. We have a manufacturing facility in Israel. We have expertise. We have R&D. We have process development. All of them are dealing and concentrating on lipids. Lipids is what we do. Our IP is in lipids. Our knowledge, know-how, everything is on lipids. We clinically validate our products and we support them with IP. We are fully integrated, which means we have research and we have all the way through manufacturing and marketing. All the people, we have them also located in the same place so we can actually talk to each other and solve problems when we are engineers and biologists and manufacturers all in the same location. We have experienced leadership as well as scientific and technological team. And why lipids? Because we believe lipids are very, very important. Lipids, we all know, are an energy source for our body. They're used for energy and they're also used for storage for energy. But lipids are also the major structural component of all the cell membranes. There are over 60,000 different types of lipids in our body. The brain, the dry mass of the brain consists of about 60% lipids. It is known today that some certain diseases and certain conditions are correlated with imbalances in lipid levels. And on the other hand, there is only limited amount of research done, which means that there is a lot more room for technology and research on lipids. So we all know one of the famous group of lipids are the omega-3 fatty acids. All started with researchers going to Greenland and studying the Inuit, and when they found out that Inuit, even though their diet is solely animal fat, had almost no death from CVD and almost no CVD and what we call metabolic disorder diseases. And soon enough, it was obvious that this paradox has to do with nutrition. Their nutrition has high levels of omega-3 fatty acids, especially EPA and DHA, which are marine-based fatty lipids. And when those Greenland Inuits move from their diet into a regular diet of the mainland of Denmark, the heart disease rate goes up, similar to Western diet. So as I said, lipids are a huge group of many different entities. This is really just a few of them. This is not all of them. And today I'm going to concentrate on the phospholipids here, basically, and to talk about the difference between phospholipids and triglycerides. So the difference between triglycerides and phospholipids lies in the structure. While the triglycerides have a glycerol backbone here with 3 legs that are each a fatty acid, and they're lipidic, they're hydrophobic, they hate water, they do not mix with water. The phospholipids have a very similar structure on one hand. They have the glycerol backbone, they have 2 fatty acids, but instead of the 3rd leg, they have a polar group here. In this instance, it's choline. It can also be phosphatidylserine when the group is serine, or ethanolamine or other. And this group is polar, which means it's charged. And this difference in structure makes it very different in function. For example, on the left here is a microscopic photo of fat cells. This is an adipose tissue. These white cells are filled with triglycerides. When we eat triglycerides, the body uses them for energy. And if we eat too much, they're stored for energy. And those cells are filled with triglycerides. On the other hand, the phospholipids are not used for energy. These are structural lipids. And you can see here that they can create, because of their very, very special structure, they are the only ones that can create this beautiful membrane structure. This bilayer will actually keep a component of water inside and separate it from the outside, and this is the biological cell. So every biological cell, plants, animals, bacteria, everything is constructed the same way with those phospholipids. What happens when we mix them with water? So this was actually done on the bench in our, the first floor in our headquarters. We went down, we asked the guys to help us out to get this picture. We mixed the beaker, the glass on the left, with some kind of fish oil, just commodity fish oil we have in the lab. And the one on the right was actually krill oil, what we had in the lab. That's, you can see the reddish color. We add it, we mix it, and we let it sit for 5 minutes, and this is what you get. The triglyceride oil will separate from the water immediately. It will not mix with the water, while the phospholipid-based oil will mix to create an emulsion. And this is a huge difference. Again, a small difference in structure, huge difference in function, physical property, and also biological function. And this leads us to the discussion on absorption. So we know that omega-3 absorption is the degree and rate at which the omega-3 fatty acids are absorbed into the living system. In this case, these are red blood cells, but not only at the red blood cells, but also the target tissues. And several studies were done. This was done by Graf and published at 2010. And this study design, they took 24 rats. They gave them a single dose of either phospholipid or triglyceride attached to radioactively labeled DHA. And after 24 hours, they sacrificed the animals, and they were looking into the brain, the kidney, and the liver. And they found out that in every case, the phospholipid attached to DHA, the DHA that came from the phospholipid diet, had an advantage, and actually the absorption was much higher compared to triglyceride. Another study was done with preterm babies that were fed with formulas that had either phospholipid attached to omega-3 or triglyceride attached to omega-3 for 5 weeks. They collected feces from those babies, and they actually calculated how much of the the omega-3 was absorbed by the babies. And they found out again that there is an advantage to the phospholipid DHA. Enzymotech also had ran one clinical study. It was run in Canada by Peter Jones, who is the director of the Richardson Center for Functional Foods and Nutraceuticals. In our study, we wanted to see the difference again between phospholipid and triglyceride-based oils, providing the same amount of omega-3 in both cases, 600 milligrams per day, for 4 weeks. And we did, because of the difference between one person to the other in metabolism absorption, we actually did a crossover study, which means the same people got all the interventions. And what we found out was, and these results are published, and you can actually read the paper. And what we saw is that in both cases of total omega-3 and EPA plus DHA, and I can tell you the same results also for EPA, there is an advantage which was clinically, statistically significant in the group that consumed the phospholipids compared to the triglycerides. And this is after 4 weeks of absorption. So why is talking about absorption or the amount of omega-3 which accumulates in the red blood cells is so important? Well, we know about this new risk factor that was published by Harris, where he looked at many studies that looked at the risk for developing either sudden death from cardiac reasons or primary cardiac disease, and correlated the amount of EPA and DHA in the blood with the risk for developing the disease. And he found out that in both cases, the lower the level of EPA and DHA in the red blood cells, the higher the risk for sudden death or for primary cardiac disease. You can see here, so this is relative risk. So this is 3.9% or 3.3, and this is 7.3. So by increasing the level of EPA and DHA in our red blood cells, we can actually increase or decrease the risk for CVD. And based on this finding, he actually developed a scale where he has two cutoffs. One at 4%. He says everybody with 0 to 4% EPA plus DHA in the red blood cells are in the undesirable zone. They have very high risk for CVD or sudden death. And all of us that are consuming, and I'm talking about myself, Western diets are somewhere around the 4%. Then there is the intermediate zone between 4 and 8%, which is something that we need to strive for. And then there is a desirable zone, and 8% is something that you will find in Iceland or Japan or Korea, places where the diet is based on fish, on a lot of marine animals. Back to our study, here we're looking actually at the change in omega-3 index. And what we found out is that after 4 weeks, when consuming omega-3 from phospholipids, you can increase your omega-3 index by 1. So if you were 4 to start with, you can now be 1. When you consume triglyceride-based oil, it goes from 4 to less than 4.5. So it seems that consuming omega-3, which is bound to phospholipid, is more efficient in increasing your omega-3 index. We added another arm to the study and when decided to again look at phospholipids, but this time cut the amount of phospholipid by half, but still have the same amount of omega-3. So we have a high phospholipid with 600 milligram, and we have low phospholipid, again with 600 milligram of omega-3. And these results are not published yet. They're not even fully analyzed yet. But what we see is a very interesting kind of a dose response. It is important to have phospholipids. The more, the better. And this is, in all cases, the same amount of omega-3. In the triglyceride, the low phospholipid and the high phospholipid. So this is very interesting, and it definitely says something about phospholipids and about the importance of phospholipids for consuming omega-3. So most common omega-3 supplements found today on the market are fish oil capsules, and fish oil production is almost always triglyceride based. The way that they are produced, it's only triglyceride. We have to remember that the initial recommendations, and even the recommendations today by the American Heart Association, the FDA, and other important bodies, is for consuming fish. And when we consume fish, it's not just triglycerides. Fish naturally have other components, and the production of fish oil leaves them out. And this part is the phospholipids. And now I would like to talk about a new product that we have, what we call the advanced fish oil, the omega PC. It's a wild fish fat extract containing omega-3 bound to triglyceride and phospholipids, similarly to the way it is found in the fish, to preserve their natural nutrient profile. The advantages, as I said before, absorption, we talked about this, efficacy because of omega-3 index, the natural nutrient profile, and this comes from the manufacturing process. So while commodity fish oil is produced using high temperature, that actually does not allow the phospholipid to remain because they're heat sensitive. The production of omega PC is solvent-based, is done in low temperatures, and actually maintains both the phospholipid and the triglycerides. Omega PC is manufactured through a proprietary process that we call the MSO, the multi-stage oil extraction, developed by Enzymotech, which preserves the natural nutrient on one hand, but removes other components such as trimethylamine. This is a volatile molecule that has that is responsible for the odor of rotten fish, and also other destabilizing molecules as well. Finally are the fishy burps. So we all know that consumption of fish oil is often accompanied by this unpleasant feeling and lack of convenience of fishy burps. And the reason comes from the fact that the absorption or the metabolism of triglycerides start already in the stomach. And that produces gases that are released, while the absorption of phospholipid is done in the lower part of the intestines, far away from the mouth, and therefore offers more convenience. And finally, sustainability. The omega PC is produced from fish that are collected through sustainable fishing, certified by either the World Wildlife Fund, the International Fish Meal and Fish Oil Organization, or the Marine Stewardship Council. And by this I ask if you have any more questions. And we are located at booth 21065, and we'll be very happy to answer questions now or have you come to our booth. Thank you.